class, this is a review for chapters 23, 25, 26, 27, and 28. This will be for exam 1, and again, this is review, so it's just highlighting, I guess, some of the main points that you really should take away from the course and what I would really like for you to study for the exam. So chapter 23 is Systematics, Phylogenies, and Comparative Biology. So we're going to look at cladograms, and comparative biology is how things, I guess, kind of compare and relate to each other, and it's also how they don't relate to each other. So here they kind of state that all organisms share many characteristics. For example, that they have one or more cells, they metabolize, which if you didn't know, this is how we know if something is considered to be living. That's if it metabolizes. So things that are not living do not metabolize. When we consider those things like abiotic, things like soil and rocks, water, those do not metabolize and therefore they're not considered to be living. Another thing is that they transfer energy with ATP, and also that they have DNA or genetic information, which could be RNA as well. And they go on to mention that life is very diverse. You can have really small organisms, and then you can have really large organisms. And so we kind of have to organize all these organisms in a certain way, and we're going to talk about how we do that. So one of the ways that we know that there are relationships, there are, I guess that some organisms have descended from others, and one of the reasons of proof is that we have fossils. Another one could be like DNA, but the fossils is actually kind of a really cool one because it's something you can actually physically see. But keep in mind that we haven't discovered all fossils, so sometimes there are gaps in our phylogenetic trees. And if we find a new fossil, then we can kind of rearrange things, but just kind of keep that in mind. It's a really popular biologist by the name of Charles Darwin, and so he kind of coined the term natural selection. And so all of that means, it says that all species descended from a single common ancestor or descent with modification. So this is actually from I think his sketchbook that he took over there at the Galapagos Islands, and that's kind of a cool thing. But right here you can see he has an early cladogram. And so I wanted to put this slide in here so that we can kind of see relationships or similarities between organisms. So at the top it says similarity may not accurately predict evolutionary relationships, meaning by off the bat, you kind of look at these two organisms and you would just automatically assume that they're really closely related. But let me give you maybe 5 to 10 seconds and just kind of look at this diagram and see if you can think of a few things or a few differences between these two organisms. Okay, so let's see if you kind of had the same ideas as me. So one of the biggest differences between these two organisms is that one is a fish and one is a mammal. Another one is that this guy has gills, and they don't have gills. Dolphins don't have gills. They have lungs, internalized lungs, and then a blowhole. Another thing that's different is the way that their tails are shaped. If you look at his tail, he kind of swims like side to side, versus a dolphin, it goes up and down. So there's kind of a few differences. The biggest one though is that this is a fish and this is a mammal. So how is it that they look so much alike, but they're not extremely closely related? And the reason for that is because they have the same selective pressures, meaning that they kind of live in the same environment. And so because they live in the same environment, they have just really adapted and evolved specifically for that environment. So that's why you see that they look similar, but they're not closely related at all. Okay, so this is a picture of a cladogram, right? And then we kind of title this as Cladistics. So characters can be an easy aspect of a phenotype. I bolded this just because I wasn't sure if you remembered this word from Biology 1406. So the word phenotype just means how an organism looks. Like if an organism has brown hair, black hair, if it has green eyes, blue eyes. So phenotype is how it physically looks. right? And so we kind of distinguish it by phenotype, and that could be through morphology, physiology, behavior, and DNA. And so I want you to kind of look at this cladogram because there's going to be one on your exam, and I really want you to kind of practice and see how you can, I guess, differentiate between these organisms. So when we read a cladogram, let me see if you can identify which one is, let's say, I guess, the more advanced animals, right? On what side of the cladogram are the more advanced animals going to be? They're going to be on the left or on the right? Right, so the more advanced animals are going to be here on the right, and then the less advanced would be here on the left. So when we're reading a cladogram, for this one, we're reading it from left to right. And so we have these kind of red lines here that kind of distinguish or bring in a new clade. And I believe in the next slide it kind of tells you what that word is. It's called a synapomorphy. So our earliest ones we have are, in this example, are sharks, right? They're fish. We mentioned that they have gills, they don't have lungs yet. And then you have a ray-finned fish. And I think the difference between these two is the skeleton. Sharks, if you didn't know, they have a cartilage, and then the ray-finned fish have actual bone. And then you could just kind of have this like steady progression, and it right here it shows you the differences. Oh, there it is, it does say it. Right, so this one, this fish is different from the shark because of this trait. This fish has a bony skeleton. And the difference between this fish and an amphibian is that this amphibian has 4 limbs. And another thing that you really need to remember when you're reading these is, like, for these synapomorphies, this means that anything past this has it, right? So the primate also has 4 limbs. The rodents also have 4 limbs. Crocodiles have 4 limbs. And the birds have 4 limbs. Birds also have a vertebrae, right? They also have the amniotic egg. So that's how we kind of read these. So anything here and on has this trait. Okay. This gives you the actual definition of a cladogram. So again, a cladogram is something that we just saw. It depicts a hypothesis of evolutionary relationships. And then this is the word that we had mentioned earlier, synapomorphy. The derived character shared by clade members. And the example that I wrote here is hair in mammals, that any mammal has hair. That's something that they all share. It's a trait that they all share. I also included this just so that you can kind of see. So these are they're both the same. They're just kind of arranged a little bit differently. But let me ask you a question. I'll give you a little bit of time to think. So can you tell me, between A, B, and C, which ones are the closest related? Which 2 letters are closely, or the closest related? So if you said A and B, you're correct. A and B are closer related than like A and C, right? Or B and C. So A and B are closer in relationship than they are to C. And so just again, these are the same, just arranged a little bit differently. So again, there will be one on your test, so just kind of practice that a little bit. I also included this one so you can kind of see the progression, like as far as evolution. This one is for plants, how plants kind of evolved out of the water. So before they were actually plants, we had what is called like algae. Algae is not considered a plant, and it's not until you get into something like a bryophyte, which an example could be like moss. That's a really common one. These are seedless plants, and then we had seeded plants, and so it's kind of like a progression. So another thing, or another way that we kind of classify organisms is by using what we call taxonomy. And so we use the like scientific names. So like if you were to think of the scientific name of a wolf, right? Yeah, you would say that that's Canis lupus, and that's considered the genus and the species. Another example here is like Homo sapien. That's the scientific name. The reason that we do that is because these are in the Latin language, which is considered to be a dead language, meaning it doesn't change. And so when we are talking about organisms, to people that maybe live in different countries, there's no issue of like using common names here that they're not familiar with, and so it just kind of alleviates any confusion. When we classify organisms, this is the classification system that we use. Again, it's called taxonomy, and so we have the most broad up here and down to the most specific. So this is the species, and then this is the domain. Domain, kingdom, phylum, class, order, family, genus, species. Some people have created like mnemonics. Like some of my students had said that they learned like Dr. King Philip came over for good soup, and so that's how they kind of memorize these in order. So if that helps you, then more power to you. So again, it was Dr. King Philip came over for good soup, or you could think of your own, however it is that you want to remember the classification system. This will be our last slide for this chapter. It's called Comparative Biology, and what I have bolded, it says, most complex characters do not evolve in one step. And so it's bolded just so that you kind of really remember that this is not a quick process, that it takes a lot of time, as well as I would even consider like luck sometimes. If it's something like a mutation, mutations are random, and the likelihood of that happening is very like small, and so it's by chance, it's by time. So for things to evolve, it's not a quick process. It's nothing that you can see in your lifetime. Something that you could see within your lifetime is like natural selection. That's more like you could see that within 40 years. But actual like evolution, that's takes a long time. And the last thing is it says initial stages of a character evolved as an adaptation to some environmental selective pressure different from that for which the character is now adapted to. And we talked about selective pressures when we saw that picture of the dolphin and the shark. Let's go ahead and move on to chapter 25, the origin and the diversity of life. So before we even had any life on the earth, we didn't even have oxygen, which is kind of a crazy concept. If you've like seen cartoons, and they'll kind of show how like the earth used to look before dinosaurs and everything, and it was just a lot of lightning, and it was just different. That's actually true. That's actually how it was. The sky and the ocean were actually a different color before the presence of oxygen, which is also something kind of crazy to think about. But a world without plants, without any life, is just, it's kind of crazy. So, but these are the gases that were in the earth before we had oxygen. So that would be CO2, nitrogen gas, water vapor, hydrogen gas, and then it says other sulfur, nitrogen, and carbon compounds. But just remember that there was no oxygen when like Earth first developed. There's a very famous experiment, and it will be on your test as well. But so it's called the Miller-Urey experiment, and they wanted to kind of replicate the Earth's early atmosphere, and so that would be without oxygen. And so this is what it looked like. 1 second, let me scroll down. And so they closed off the ends so that no oxygen, right, could go in, and so they heated water to simulate the ocean, and then they added sparks, which are right here. They added electricity, like sparks, because as we mentioned earlier, there was a lot of lightning within the early, I guess, Earth, and so what they found at the end of this process was that it made amino acids. So from this process, they did not make life. That was kind of like a controversy that they had created life. They didn't create life, but they did create the building blocks of life, which are considered to be the amino acids. So when we actually did have life, it originated out of the ocean, and these are going to be really small organisms known as bacteria, and so I have it highlighted there, right, that the single-celled organisms are the first life forms on our planet, and so they originated in the, I think they're called thermal vents, which are at the very bottom of the ocean where a lot of hot gas comes out. And so these early bacteria are the ones that actually were able to photosynthesize. So some bacteria can photosynthesize, and it's because that they were photosynthetic that created the Earth's early oxygen. So if it wasn't for this type of bacteria, which by the way it's called cyanobacteria, and I don't think I have it here, but I can put it here. Cyanobacteria. So that's a photosynthetic bacteria, and it's the one that produced Earth's early oxygen. If it wasn't for that bacteria, we wouldn't be here. Plants wouldn't be here. Nothing would be here without this bacteria, because it made the oxygen for us. I just mentioned this, but it says, seems likely that Earth's first organisms emerged and lived at very high temperatures. And so again, that would be in the thermal vents of the ocean floor. And it says here, like, when that happened, 3.8 billion years ago. And so how do we know that this is true? Because we have microfossils that kind of prove that. And so now we're going to talk about climate. So the difference between climate and weather is that weather is daily and climate is something that's on a yearly basis. And so it says here that dramatic shifts in all of these factors led to mass extinctions influencing the course of evolution. So I'm sure you all are aware, like, there's like eras in geologic time, and so it's mostly driven by climate. And so when there is a big climatic shift is when you'll have a lot of organisms kind of die off, and then you'll have this surge of different organisms that come from that. Right here I have a eukaryotic cell. We know that it's a eukaryotic cell because it has a nucleus. Prokaryotic cells don't have a nucleus. But so the main takeaway that I really need you to know about this is that eukaryotes have this thing called that word is hard for me to say. Compartmentalization. There we go. Which means that they have kind of like these organelles that are a little bit more complex. So things like the Golgi apparatus, the nucleus as well, endoplasmic reticulum. So things like that are considered a compartmentalization of cells, and so that's unique to only eukaryotes. That's nothing that you will find in prokaryotes. Another thing that I need you to remember is the endosymbiotic theory, and this is specifically to eukaryotic cells. And so this is actually like a proven thing. It's not like a question of we're not sure if this happened. So what happened was that we had a eukaryotic cell, and it happened to engulf two types of bacteria. One is the cyanobacteria that we mentioned earlier, so this is a photosynthetic bacteria, and then a aerobic bacterium, which is able to do cellular respiration. And so if you can kind of guess what they are, I mean, the colors might be a giveaway. So I have something that's green and red, and so what are they? This is actually the mitochondria and the chloroplast. So we know that this is true because the chloroplast has its own DNA, and that DNA is different from the DNA found in the nucleus. Same with the mitochondria. Mitochondria has its own DNA, and it's different from the DNA that is found inside of the nucleus. So this process is completed by endocytosis, where it engulfs these two cells, and then they kind of live there forever. And so once that happened, now the cell divided with replicated, and so it's just kind of passed on from there. But again, that's the endosymbiotic theory, and that's something that happened in eukaryotic cells. Moving on to chapter 26, these are viruses, and so the main thing that or maybe one of the bigger things that we need to remember about viruses is that they are non-living. We might actually be more familiar with viruses than we'd like to be due to like COVID-19. But so you may have a little bit more knowledge than you may have thought you had before. But again, viruses are non-living. And I have highlighted like how do we know that they're non-living. One of the reasons that they're considered to be non-living is that they can't metabolize. And so I have it here that they don't have a cytoplasm. And what a cytoplasm is it's like that jelly in the cell that kind of just, it keeps things together, but it also has another really important role that isn't talked about, I think, as much, but it actually is a process that happens there, which is called glycolysis. And glycolysis is part of cellular respiration. And so this is a form of metabolism, right? So metabolism does occur in the cytoplasm, and because the virus doesn't have a cytoplasm, It does not metabolize, therefore we consider viruses to be non-living. But they do have other things that I guess are found in all viruses as far as their structure. They do have DNA or RNA, single, double, and you can have an RNA virus, a DNA virus, or a retrovirus. But the big things that I really need you to remember there are the bolded ones. This slide I like because it talks about but shows you the differences in size. So these are the viruses, these little ones here in green, and then this is also viruses. And so notice their size compared to a bacteria cell. So this is a bacteria cell, so they're really small, right? Viruses are very, very small. This is another bacteria, and then this huge thing right here in green is a eukaryotic cell. So in comparison from eukaryotes to viruses, viruses are really, really small, which is why you can't see them on the surface like of a table. You can see bacteria cells, like I know you can like spray something and then like use a black light and you can see like bacteria. You wouldn't be able to do that with viruses. That's how small they are unless you had a microscope. So just kind of keep that in mind. So that's why like with COVID, when people like sneeze and all of that, like viruses are so small and they can travel through the air and we can't really see them, and so that's why it's a little bit more like scary in a sense. As far as their structure, they have what's called a capsid, which is the like most outer layer. And so I don't know if I have a picture somewhere. I guess we can go back to this one. So they all have kind of this outer layer called the capsid, and it's called like a gelatinous sheath. And so I need you to kind of remember that it has a protein sheath or a capsid, and that's just used for protection. It's also used to kind of hide. So if they have this like jelly outer layer, they can kind of be undetected among other cells. So how is it that viruses are able to replicate if they're not alive and they don't metabolize? And I'll show you a picture in a second. But so what they kind of do is they will inject their DNA into another cell, and that's how they're able to replicate. But this doesn't happen. Losing my words, sorry. But viruses, and I have it somewhere written that they're very specific, off right there in the middle, excuse me. Viruses are very specific. They cannot just attach to any cell like the COVID-19 virus, right? It doesn't infect like all of your cells in your body. It's really specific to the respiratory system. So only certain cells within your body are the ones that are attacked by COVID-19. Same with different viruses, they're very specific. So if they do not have the surface receptors, that virus cannot latch onto the cell and therefore it cannot infect that cell. So viruses are very specific. They won't just infect every cell. They only target specific cells. And something that they have noted here is that they can be virulent or they can be dormant for a long period of time. And so here's a really good picture that kind of shows you some of the ways that they can replicate. So they will latch onto a cell and then it'll inject its own personal DNA into a cell to recombine. And so let's kind of review what DNA is. So DNA is not like a living thing. The only thing that DNA is instructions. It's instructions to make things, instructions to make proteins, to make mitochondria, to make cytoplasm, whatever. Right? It's just instructions to do things. But so what will happen is that a virus will change the instructions. Instead of making what it needs to make, now it's going to give the instructions to make baby viruses and then just kind of spread viruses everywhere. And so that's why maybe you've heard the term that viruses like hijack a cell, and that's true. That's what happens. So instead of the cell needing to make what it needs to make, it's going to change the directions, and instead it's going to make baby viruses. Another one here, the lysogenic cycle. This is the one that they had mentioned earlier, that it kind of just stays dormant for a long period of time. And it's kind of like a slower process. So it really does depend on the virus if they are more, I guess like this, more aggressive or not as aggressive. One very popular virus that's actually really prevalent in the Rio Grande Valley, and we're trying to combat that by educating and bringing more awareness to it. And HIV is something that really affects your T helper cells, and so Some of the, I know like it, so some people have like a really, they're really scared of HIV, and I understand why they would be scared of HIV. But just know that if you do happen to catch HIV, you can take a pill and that will help you not get AIDS, which is something that really hurts your immune system. And so what that really does is that it kind of stops the virus from replicating. And so this picture just kind of shows you how that medication will stop the process of those viruses hijacking the cell and just making copies and copies and copies. Moving on to chapter 27, prokaryotes. So prokaryotes are prokaryotic cells. They are considered cells, so they are considered to be living. They're considered to be the most simple cells. These are also the first type of cells that we mentioned earlier, first types of cells ever to appear on the Earth over 1 billion years ago, before eukaryotic cells appeared. And they fall under two domains, which are the bacteria domain and the archaea domain. We've got a simple diagram where we have the most recent descendant, which is the eukarya. So these are the eukaryotic cells, and these came from our bacteria domain and our archaea domain. Excuse me. Some characteristics of prokaryotic cells is that they are unicellular. Their cell size, which is really, really small, but it's not as small as viruses. They have what we call a nucleoid region. So they have no nucleus. And as far as cell division, they do something called binary fission. So they do not perform mitosis or meiosis. They divide by binary fission. So these are kind of the main things I really need you to remember about these prokaryotic cells. As far as their shapes, I'm not sure why they have it laid out this way, but the three shapes are bacillus, coccus, and spirillum. So bacillus is rod-shaped, coccus is spherical, and the spirillum is helical-shaped. And so they have the shapes here: bacillus, which is rod-shaped, coccus, which is spherical, and the spirillum, which is the helical shape. So those are the three shapes of prokaryotic cells. Prokaryotic cells have peptidoglycan, which is, let's hold that really quickly. So peptidoglycan is proteins, and so some of the prokaryotic cells have more peptidoglycan than others. And so we distinguish those between gram stains. So if it has a lot of peptidoglycan, it says here, right, gram-positive bacteria have thicker peptidoglycan, and they stain purple. So gram-positive purple, so if you remember, right, P, positive purple. So gram-positive is purple. Gram-negative has less peptidoglycan, and it's going to be pink. So why is that? Why is one purple and one is pink? So they stain them both with a purple color, but one isn't able to absorb the purple color as much, and so that's why this pink is actually it's like a faded purple. So gram-negative has less peptidoglycan, so that means that it doesn't have as much protein in its cell wall. Gram-positive has more protein, which means it has a thicker peptidoglycan cell wall, and it stains purple. So let's kind of look at this a little bit further. So these are both two bacillus, right? They're rod-shaped bacteria cells. They kind of show that they cut a chunk out of the cells, and so this is what it looks like, really big. So one of them, this one, the gram-positive, is the one that has a thicker peptidoglycan layer, and it shows you these rods are the peptidoglycan. And then the gram-negative has more fats. So the phospholipid bilayer is lipids, and lipids are fats. So because it's not as protein rich, this one has less peptidoglycan and it stains pink. So this is how it would look under the microscope. So this is a pink one, so we know that this is gram-negative, and then this is purple, so this is gram-positive. Some of the ways that they can reproduce, it says here that they don't reproduce sexually, so this means like they don't undergo meiosis. So some of the ways that they can get genetic, I guess, variation is through conjugation, transduction, transformation, cell-to-cell contact, by viruses and by the environment. Another one that they, I think, didn't leave in is like a mutation. Mutation can also bring in more genetic variation, but I guess this is specific to how they reproduce. But just remember that mutations also bring genetic variation. This is our last chapter for the review. We are almost done, guys. So this is chapter 28, Protists. And the Protists are actually a kingdom. And I know this picture is kind of pixelated, but I like it because it kind of shows all of them. So if you notice, they're all very different. So I have all these different types of cells. And so why is it that we have a really diverse kingdom and we put them all together? And the answer kind of is like that we don't really know where to put them, right? They're not a fungus, they're not a plant, they're not an animal. So because they're not either of those, because they're not a fungus, plant, or animal, we put them all in this kind of like just kitchen sink category, which is called the Protist kingdom. So I have it highlighted here. Like, why are they Protists? Because they're not fungi, plants, or animals. So they are a very diverse kingdom. And they're so diverse that they can either be unicellular or multicellular or in a colony. They can be big or small. They have all types of symmetries and all types of nutrition. The ways that they can move by using their flagella, cilia, or pseudopods. So we know flagella are different because they're kind of, they're a lot longer and you won't have as many flagella on a cell. Cilia is more hair-like. So they're really short and there's a lot more of them. Pseudopods, you find them in amoebas, and I'll show you a picture of what that looks like. A little while ago we mentioned nutrition, so this is how they make their food or how they eat. So an autotroph is something that makes its own food, and so they do that by being photosynthetic or chemoautotrophic. Or if they cannot make their own food, we call them heterotrophs, and so that means that they have to like eat their food, just like how we do. We have to like eat food. We can't just make our own food. And so those would be considered phagotrophic. Because they're so diverse, they can reproduce in multiple ways. It can be either asexual or sexual. So some of the examples include mitosis, budding, and then some of these it says meiosis is a major in eukaryotic innovation, so they can even use meiosis. And so we're almost done. I'm going to show you a few examples of some protists, and anything that I have like highlighted or on these cells, I really hope that you write it down and kind of practice it because it will be on your exam. So the first one is a Euglena. A Euglena is a protist that is photosynthetic, and we know that it's photosynthetic because it has a lot of chloroplasts, and so we kind of coined that term phototrophic. So if it's phototrophic, that means that it's able to photosynthesize and it has a lot of chloroplasts. So here it has them right that it has, so we can actually count these. 1, 2, 3, 4, 5, 6, 7, 8. I think I counted that right. So this one has 8 chloroplasts. So again, it's a photosynthetic protist. It also has a flagella, so this is how it moves. It has a really, really big flagella. Where do you think you would find something like this? This would be in fresh water, like a lake, a stream, canal. You could find this here, like in the Rio Grande Valley, you could find these. Same with the Paramecium. You could also find these in like pond water or canal water here in the Rio Grande Valley. We call them Ciliates because they have a lot of cilia. So you see these little hairs here. This is a lot, so we call it a ciliate, but specifically this is called a Paramecium. Some of the things about this is that it has 2 nucleus, the macro and the micronucleus. Another thing is that it has a contractile vacuole, which I don't see. Okay, here. Contractile vacuole. That's unique only to ciliates, and that's how they're able to either take in or extract water that they don't need anymore. Like if the water is like really salty, that's how they're able to adjust. Another one is algae. So people always kind of assume that algae is a plant, but not all algae is a plant. Some is actually a protist. Diatoms is another example. So these are also photosynthetic. unicellular. And the last one that we will leave on is the amoeba. I've only seen an amoeba like in person once, and it's actually really cool just because they move really strange. So if you can like YouTube a video of amoebas, I would hope that you do, because so you can kind of see how they move. But so these have the pseudopods, and I don't see it here on this slide, but it's kind of added so you kind of remember the word pseudo. I feel like I'm missing an E. Pseudopods. Alright. I'm going to double check that I spelled that right. 1 second. Okay, guys. Okay, cool. So it has pseudopods, which are these, and so we call them false feet, and that's how it moves. It's able to use them to move, and as well as it can like engulf organisms, which is how this eats. It kind of gets its pseudopods and wraps it around something and then just engulfs it, and starts to digest it. But yeah, so that is your review for exam 1. If you have any questions, feel free to send me a message on Pronto or send me an email.